Hey guys, welcome to my YouTube channel. This is Pinky Sense Academy. Please like, subscribe and comment. This is Biology Form 1 revision questions and answers. It's an end of term 2 exam. So the first question, you're asked to state 2 scientific skills imparted to a biologist. Scientific skills are the ones that you learn when you are doing a biology course, like drawing, recording data, analyzing data, interpreting data, etc. State 2 professions open to a biologist. The professions are the careers. You can become a biology teacher, you can study medicine, pharmacy, become a pharmacist, a doctor, yeah, all those. The diagram below shows one of the characteristics of living organisms. Identify the characteristics. In this photograph, you can see clearly that we have this animal that is coming out. So in short, this animal is reproducing. It is giving rise to a live young one. So it is the characteristic displayed there is reproduction. Then you asked what is meant by the term taxonomy. We say taxonomy is a science of classification. The scientific name of a dog is Canis familiaris. Write the name correctly. Now, this being a scientific name, the genus name should start with a capital letter, species name should start with a small letter. That's why I've written them like that. And then, since they are scientific name, they should be underlined separately. That's why I have underlined Canis and familiaris separately, because I'm not typing. Identify the genus and species name. So the genus name is the first name, which is Canis. Species name is the second name, which is familiaris. Then we have this diagram here. You are told that the figure below represents a specific cell organelle. It's supposed to identify the cell organelle. The cell organelle there, it's a nucleus. I know some of you have never seen such. So it's a nucleus. And you are also asked to explain how the cell named here, explain how the cell named in one above is adapted to its function. It has a nuclear envelope to protect the DNA. Mainly the nucleus stores the genetic material, genetic information in form of DNA. What is the function of the part labeled A? Part labeled A here is the nucleolus, and nucleolus is involved in production of ribosomes. Identify part labeled B and C. Part labeled B here, and also part C. So part labeled B, they are the ribosomes. You can see they are located on the endoplasmic reticulum. The endoplasmic reticulum that has ribosomes on its wall is rough endoplasmic reticulum. And then C is the nuclear pores. Name the organelle that perform each of the following function. Osmoregulation in amoeba. Osmoregulation is regulation of the amount of water in an organism. So in amoeba, you find that they have temporary vacuoles or contractile vacuole, which collect water or other fluids in them. And then when they are full, they fuse with the membrane and get rid of that water. So osmoregulation is carried out by the contractile vacuole. The organelle that carries out digestion and destruction of worn out cell organelle, they are the lysosomes. Lysosomes. Then the next question, you're given the diagram of a specialized cell in a human being. So this one is a sperm cell. clearly from the parts. It has a tail, it has the middle piece, and then it has the head, which has R chromosome in it, and the nucleus. So you are supposed to identify the cell. The cell, of course, it's a sperm cell. And then you name the parts A, B, and C. Part A, part B, and part C. Part A is the acrosome. Part B is supposed to be the nucleus. Part B is the nucleus because it's there. And then part C is the middle piece that is normally made of mitochondria that acts as source of energy. State the function of the part labeled A. Part labeled A here, it's the acrosome. So it has lytic enzymes that destroys or break down the membrane of the egg or ovum so that the sperm can fuse with the egg. State 3 properties of the cell membrane. State 3 properties of the cell membrane. Semi-permeability, that means the cell membrane selectively allows substances in and out of the cell. Sensitivity to changes in temperature and pH. It is sensitive to changes in temperature and pH, and then it also has electrical charges, or we say it is polarized. It has positive charges and negative charges. So during practical class, Form 4's estimated the field of view to be 3.8 millimeters. Using the low power objective lens, they observed Spirogyra cells across the same field of view and counted 8 cells. Calculate the size of the cell and give your answer in micrometers. So the first thing that you're supposed to do there, you convert the millimeters into micrometers. So 1 millimeters is equals to 1000 micrometers. How about 3.5 millimeters? So you cross multiply, you get 3.5 millimeters multiplied by 1000 over 1 millimeter. So that millimeter units cancels out, and then you get 3500. So to calculate the size of the cell, you use this formula. Length of the cell is equals to the diameter of the field of view divided by the number of cells across the diameter of the field of view. Now that you have 3500, you divide by 8. That's why you get 437.5 micrometers. So the first thing, first of all, you convert the units into micrometers, and then you use this formula. Length of the cell is equals to diameter of the field of view over the number of cells. 3500 divided by 8, you get 437.5 micrometers. The next question, you are told to distinguish between the following terms: magnification and resolution of a microscope. So magnification, it is making very small organisms to appear bigger so that they can be seen. That is to magnify or to enlarge. Resolution is the ability to distinguish between two structures that are close together as distinct entities. That means two close points, or 2 points that appear so close together, then the microscope is able to separate them so that they appear separate. That is resolution. So microscope has those two functions: magnification, that is enlarging, and resolution, that is making two close points appear separate. Then mounting and staining of the specimen, where mounting is putting the specimen on a slide in the appropriate medium before covering it with a cover slip so that you can observe under a microscope. Staining is making different parts of the cell more distinct or more clear by using stains, where you apply stains, for example, iodine solution or methylene blue. That one will allow the cells to be very clear. So you stain for clarity. Mounting is just putting the specimen on a slide, and then you put it, or you put a cover slip on it, and then you put it on the stage of a microscope for easy observation. What are the following procedures done? Why are the following procedures done when preparing sections to be observed under a light microscope? This is a very common question, even in KCSE. So making thin sections, that is to allow light to pass through. Using a sharp razor blade to make the sections, it is to ensure that the cells are not distorted. Adding water, you make them turgid to make the cells turgid or even prevent dehydration. What do you understand by the following terms? Anatomy. It is a study of internal structures of living organism. That is anatomy, study of internal structures of living organisms. Excuse me. Biochemistry is the study of chemical changes inside the organism. Biochemistry. Number 6, you've been given a microscope there. That's a figure showing a microscope. You are supposed to identify the part labeled C, part labeled E, and F. Part labeled C, this one, it's a fine adjustment knob. Part labeled E, here, it's the revolving nose piece. Revolving nose piece. I think I did not label it. Let me just label it for you. Part labeled E, revolving nose piece. Revolving nose piece. And then part labeled F, it's the objective lens. Then you're asked, what is the role of part labeled A, B, and G? Part labeled A, that is the eyepiece lens. It contains lens which contribute to magnification, or it helps to enlarge an image. Part labeled B is the coarse adjustment knob. It normally brings an image into rough focus by raising and lowering the body tube. And then part labeled G, which is the condenser, it concentrates right onto the stage. So microscope is also a very, very common question, so you should be careful about it. You'll see more in the videos. State 2 precautions that should be taken when storing a microscope after laboratory exercise. You should always clean that microscope and store it in a safe place, away from moisture, away from dust, because when you expose it to moisture, it is going to rust. State 3, physiological processes that are involved in movement of substances across the cell membrane. Of course, you have diffusion, you have osmosis, and also active transport. Active transport is also present as a movement of substances. So you can add there active transport. So potato cylinders were weighed and kept in distilled water overnight. They were then reweighed. So this is before and then after. Before you have A, B, and C, and then after. So you are asked to explain why the mass of the potato cylinder had increased. Of course, when you place those potato cylinders in distilled water, water will get into the potato cylinder by osmosis, therefore making them big or increase in size. State 4 factors that determine the rate of diffusion of materials. Temperature. When the temperatures increase, the rate of diffusion increases. Thickness of the membrane. The thicker the membrane, the lower the rate of diffusion. Size of the molecules. We find small sized molecules move faster or diffuse faster than large sized molecules. And then there is diffusion gradient, where when there is a a big diffusion gradient, that is the difference between a concentration of two molecules, then you find that the rate of diffusion increases. State 3, adaptations of the leaves to photosynthesis. State 3, adaptations of the leaf for photosynthesis. The leaves, they are thin to reduce the diffusion distance for the gases, also to allow sunlight to penetrate through. They have numerous chloroplasts to trap sunlight for photosynthesis. The leaf also has xylem vessels a short distance away to supply water and mineral salts to the photosynthesizing cells. And they have phloem a short distance away to transport the manufactured food. The leaves also have palisade mesophyll cells that are closely packed to trap maximum sunlight. State the function of each of the following tissues. Xylem are the vascular tissues where they are made of xylem and phloem. Xylem transport water and mineral salts to the photosynthetic cells. Phloem transport manufactured food. Epithelial cells, it protects the inner tissues of a plant from mechanical damage. Blood tissue, it protects the body against diseases and also transport materials, e.g. oxygen, metabolic waste, and nutrients. Meristematic tissue, they actively divide to bring about growth, where we have the apical meristem, bring about increase in height and of the shoot and root, and lateral meristem brings about secondary thickening. The next question, you are asked to distinguish between turgor pressure and osmotic pressure. Turgor pressure is the outward pressure produced by the cytoplasm and its content pushing towards the cell wall, while osmotic pressure is the force that is developed by a solution to draw water through a semi-permeable membrane. Plasmolysis and deplasmolysis. Plasmolysis is the shrinking of a plant cell when placed in a concentrated solution, while deplasmolysis is when you place a plasmolyzed cell in water to make it turgid. That is deplasmolysis. You place already shrinked cell into distilled water to make it turgid. The next question, now that is page 9. So you are told that the figure below shows an organelle that is found in a certain cell. You study it and answer the questions that follow. You identify the cell organelle, that's chloroplast, and then you're told to identify part labeled E, A, C, and B. So part labeled E is the lamellae, part labeled A is the granum where light stage of photosynthesis take place, part labeled C is the starch granule, and part labeled B is the stroma where the dark stage of photosynthesis take place. Then we move on to this question. You are asked to write an equation for the reaction that takes place at A. A is the granum, where it's the light stage of photosynthesis takes place. Water molecule is broken down into oxygen atoms and hydrogen ions. B, carbon dioxide fixation, that is in the stroma, where carbon dioxide is combined with the hydrogen ions to form a simple carbohydrate or a simple sugar. Apart from glucose, name two other substances that are formed in the stage of photosynthesis that takes place in B. There is formation of water, is formation of proteins, sucrose, lipids, everything is made from the glucose. Glucose can be converted into sucrose, can be converted into starch, so many things are made from there. Form 1 students going for an excursion at Amazon rainforest required to correct specimen for running in biology. Name the apparatus that can be used to correct the specimen. This one is a big organism. You can only correct it using a bait trap because it looks like a rodent, a small rodent. Next question. You are told to outline 2 ways in which the knowledge of biology is applied in agriculture. It's applied in crop rotation and also pest control. Explain how the surface area to volume ratio affects the rate of diffusion in living organism. So the higher the surface area to volume ratio, the higher the rate of diffusion. Organisms that are very tiny or very tiny molecules have a large surface area to volume ratio, while big organisms or big molecules have a lower surface area to volume ratio. Therefore, that means that the smaller the molecule, the higher the rate of diffusion. State why the following processes are essential in living organisms. Excretion, it helps to remove metabolic waste. Irritability, it helps an organism to respond to changes in environment. You are given a diagram here. You're supposed to study the illustration A and B. You can see A has water only, B has salt particles. In the end, the water molecules have reduced. In A here, they are very minimal, where in B, it has increased. Now you are asked, name the substance responsible for the change there. The substance responsible is water, or you can also say partially permeable membrane, the dialysis tubing. Account for the change observed. Water molecules moved from a region of high water concentration or potential to a region of low water concentration across the semi-permeable membrane, and that is why the water molecules decreased while the other solution increased. Name 3 aspects of light that affect the rate of photosynthesis. There is an aspect of light intensity, light duration, and light quality. 3 activities of the cell that is controlled by the nucleus. It controls protein synthesis, it controls enzymes synthesis. Thank you guys for watching. Please like, subscribe, and comment. Watch my playlist and my podcast, Pinky Science Academy, and playlist Pinky. It has every revision need for your biology and chemistry course. See you in the next video.